super grateful for the opportunity to share with you my work looking at the role of diet in influencing patterns of venom evolution in cone snails. The use of venom for predation has evolved several times across disparate animal lineages, including snakes, snails, and spiders. And venoms are typically composed of complex mixtures of toxic proteins. And the extensive variation in venom composition is documented at nearly all biological scales of organization. And so a major goal in venom biology is to try to understand what are the factors that shape and influence all this variation that we see in nature. Diet is thought to be a major ecological force governing patterns of venom evolution because venom is intricately linked to a species' ability to capture and apprehend prey. And there are currently two major hypotheses that are thought to explain how diet should impact patterns of venom evolution. And the first is that diet should predict what types of proteins a species should employ in its venom cocktail, because natural selection should shape the venom repertoires of species to be able to take down the physiologies of their particular prey. And so this essentially suggests a covariation between diet and venom components. So for example, snakes that feed on different diets, vertebrates or invertebrates, should have different venom compositions. And similarly, species that share similar diets should converge on their venom composition because they're optimizing on similar prey species. The second hypothesis is that dietary breadth should be positively correlated with venom complexity because you need a greater number of venoms in order to take down a wide variety of prey species. And although rarely invoked in venom studies, this prediction is largely consistent with greater, broader ecological and evolutionary thinking about trait diversity and niche breadth, such that in 1965, Van Balen came up with the niche variation hypothesis, which posits that individuals or populations with wider niches should be more diverse, phenotypically variable than populations or individuals with narrower niches. And so there are 2 points I want to make here. The first is that these two ideas are non-mutually exclusive. They just simply describe different ways in which we expect diet to influence patterns of venom evolution. So the first hypothesis makes a prediction about what types of venoms we should expect in a species cocktail, and then the second hypothesis makes a prediction about how many venoms we should expect. And the second point is that both of these hypotheses have been tested in the literature, and sometimes you find support for these ideas, and sometimes you don't. But the majority of studies test these ideas looking at among population variation in venom composition within species or among very closely related species. So very rarely are these hypotheses tested using species-level variation across a radiation of venomous organisms. And without these broad comparative studies, it is difficult to discern between the noise that's inherent within the particularities of a given study system versus the actual evolutionary trends in venom evolution. And so that's the gap I tried to fill in my study in cone snails. And so you might ask, why snails, Mark? And my answer to you is that they're awesome. But in any case, these snails are cone snails. They're predatory marine snails concentrated mostly in the tropics, reaching over 800 species. Cone snails are predatory and carnivorous, and they feed on either worms, mollusks, or fish, but a small combination of species feed, a small minority of species feed on combinations of these three. Cone snails are venomous and produce a cocktail of venomous neurotoxins that's produced along the venom duct and is injected into the prey with a radula that's modified to be like a harpoon or spear. So radula are analogous to teeth in snails. And so to test those dietary ideas, I generated venom duct transcriptomes for 12 species that span the phylogenetic distribution of cone snails. Ecologically, these 12 species represent 10 worm eaters, 1 molluscivore, and 1 generalist that feeds on worms, mollusks, and fish. And I paired this venom composition information with ecological data from the literature to test those dietary ideas. And so the first hypothesis, that diet should predict what types of venoms a species should employ. And so from my sampling, I have these 3 dietary classes that are recognized in the cone snail venom literature. And it is predicted that these diet classes should be related to what gene superfamilies should be expressed in these cone snails' venom cocktails. So in cone snails, venom composition is characterized by the relative contribution of these different gene superfamilies, and there's named like the A gene superfamily and the B gene superfamily, and these are thought to be optimized to take down these particular prey classes. And so what I can test with this sampling is that the vermivorous species should be more similar to each other in venom expression relative to the molluscivore or the generalist. So to test these hypotheses, I first built a phylogeny from over 800 common cellular genes that I discovered through the transcriptome sequencing. It's time calibrated, pretty well resolved. Those values at the nodes are bootstrap support, and those black circles are 100% bootstraps. And the species names are colored by diet. Black is vermivore, orange is molluscivore, and green is generalist. Across these 12 species, I discovered 58 gene superfamilies, 14 of which I list down here that were at least 10% of the venom composition in at least one of these species. And so here's a heat map showing you venom composition and the relative contribution of these different gene superfamilies. And so the heat map goes from white, or 0% of the venom composition, to over 30% in dark blue. So for example, Conus radis expresses the L gene superfamily, and it represents over 30% of the venom composition of this species. And so as you're looking at this and trying to relate it to the diet colors, and it's sort of like all over the place. There's a lot of diversity, extensive variation, and it seems like each species is doing its own unique thing. And when you look at any of these vertical columns, sometimes a gene superfamily is very important for that species, it's a major part of the venom composition, and sometimes it's not. So to put this in a more quantitative framework, I borrowed the ecological similarity statistic, shown as D, which was originally used to quantify niche overlap in habitat and diet in anolis lizards, and used this to calculate the amount of similarity in venom composition across all possible pairwise comparisons across my 12 species, and categorize each comparison based on whether or not it was between a generalist versus a vermivore, a molluscivore versus a vermivore, or a vermivore versus a vermivore. And so you can think of this as different diet, same diet, with the expectation that species that have similar diets should have similar venom compositions. And on the y-axis, I have that p-value going from less similar, smaller values to more similar, higher values. And here are the distribution of those d-values. So after correcting for phylogenetic relatedness, and ANOVA found no significant differences between these three categories, suggesting that diet does not predict the similarity of venom composition patterns among these three categories. And you can kind of see this already from the distributions, where sometimes when you compare two vermivorous species, they're more different to each other than when you compare those same two species to a molluscivore or a generalist. And so why might this be? And the first explanation I can give to you is biological. So whenever you measure the molecular evolutionary rate of venom genes, they're among the fastest evolving genes known in animal taxa. So perhaps this high propensity for change leads to evolutionary trends in divergence in venom composition rather than convergence among species that share similar diets, which remains the dominant expectation in venom biology. The second is that perhaps the way in which venom and diet has been categorized and classified in the cell literature for the past decade is inappropriate for estimating the evolutionary dynamics between those two parameters. So for example, on the venom side, when you're making a statement about this hypothesis, such that diet should influence venom evolution, you're essentially making a statement about how venom proteins are evolving functionally to target the physiologies of particular prey species. But the way in which venom composition is described in cells by venom gene superfamilies, these gene superfamilies provide little to no predictive information on protein function. So essentially, characterizing venom composition in this way is meaningless because you don't capture the functional aspect of what these venom proteins are doing. On the diet side, if this is most clear when you call something a vermivore or a worm eater, the word worms means a lot of different things. And so this is a chronogram showing the origination of major worm lineages in the fossil record. And the arrows are pointing to lineages of worms that snails feed upon. And these lineages have been diverging from each other for over hundreds of millions of years. And not acknowledging all of this diversity likely obscures actual evolutionary trends between diet and venom. Second hypothesis, that your diet diversity should be correlated with how complex your venoms are. To do this, I obtained dietary diversity metrics for 10 of the species in my study, and this came in the form of Shannon's diversity index, or the H index, which takes into account the number of species a cone snail preys upon, and the relative frequency at which it preys on those species. And so that's on the y-axis here, going from less diverse, lower values, to more diverse, higher values. And on the x-axis, I have the number of unique mature toxins that I discovered in each transcriptome. That was my measure of venom diversity. And so here's the result. Every point here is a species, and after using a phylogenetic generalized least squares analysis, I found a significant positive relationship between these two parameters. If you're wondering about this point way up here, that's Conus californicus, and it's already thought of as an outlier in the cone snail venom literature. And when you remove that point, this relationship becomes stronger and more significant. So I'm pretty confident in the signal of this result, and what's nice about this is that so far, every time it's been tested in cone snails, we find this relationship. So this relationship is true among populations within species and also within an individual's lifetime. And furthermore, this is consistent with broader ecological and evolutionary thinking on trait diversity and niche breadth, so that niche variation hypothesis. And so, one question you might ask after getting this positive result is, what's the significance of this? What are the evolutionary consequences of this relationship existing in nature? And one cool thing about cone snails is that there's a really simple relationship between genotype and phenotype, such that the genes that code for venom, for the most part, code directly for the venom protein. So that you can think of this as a relationship between dietary breadth and the number of venom genes that are expressed or available for selection. And what's cool about this is that I've already been trying to look at venom gene diversity variation in the genomes across several hundred species of cone snails as a way to measure the intrinsic capacity for organisms to diversify. So linking to ideas about evolvability, grounded in the logic that if you have more genes coding for this ecologically relevant trait, venom, then you have more opportunities to potentially generate novel phenotypes that may be important for selection in the future. And this perhaps might be correlated with higher level biodiversity patterns such as speciation rates or rates of phenotypic evolution related to the venom apparatus. And that dietary breadth result I just showed you suggests that we can use cone snails as a system where we can address the evolutionary consequences of being a specialist or generalist on that dietary breadth spectrum and what impacts it has on these higher level biodiversity phenomena. So that's where my research program is going. And thank you for listening. But in the sake of winning evolution bingo, may the force be with you for a wonderful conference. Yes, so functionally,
repeat the question. Yeah, we couldn't hear the question.
Oh, is there evidence that the venom cocktail is optimized to take out particular prey species, right? And so have other studies shown that? And the answer is yes. So when you have a common diet or if you have species that vary in diet, and then test the venom cocktails of each of those species on sort of like one particular prey, then you get the relationship that you expect is that species that tend to feed more on that model organism, that you're